Welcome to SSR Pharma Media, your trusted source for pharma knowledge. Today's series Phytopath Quiz Biosynthesis Unlocked Biosynthetic Pathway. In that, we are going to discuss about biosynthesis of glycosides. Prepare, practice, succeed. So, in previous video, we have discussed about cyclic acid pathway, means aromatic biosynthesis. So, today we are going to look over biosynthesis of glycosides. So, and remaining biosynthesis of alkaloids, biosynthesis of isoprenoid compounds, biosynthesis of amino acids, we will see in forthcoming videos. So, we will discuss about the glycoside formations. So, what is mean by glycosides? So, glycoside is the combination of glycon part, that means sugar part, and aglycone part. So, it's containing glycone part, so sugar molecules, and aglycone part, it is a non-sugar moiety. And in this linkage between oxygen means O-glycosidic linkage. So, in nitrogen group means N-glycosidic linkage, and S-cyanide group means S-glycosidic linkage. So, depending on the atom linking them. And what is the roles and functions of this glycoside? So, first is sugar part of the glycoside is a aglycone part. So, increase the water solubility of the molecule, helps in transport and storage in plants, determines specificity of enzymes hydrolysis and how the glycoside is broken down. So, it is the roles and of or functions of the sugar part, that means aglycone part. So, non-sugar part, so it is also called as, this aglycone part. So, non-sugar part is a aglycone part. It is also called as xenin. It's a aglycone part, it's a non-sugar part, it's a xenin. So, the functions of this non-sugar part, aglycone part, is responsible for biological activity and pharmacological effect. And also it is used for the determine the therapeutic effect or toxicity effect, and released when the glycoside is hydrolyzed by enzymes or acids. So, these are all the functions of aglycone part. So, next, so types of glycosides, there are 4-5 glycosides are there: cardiac glycosides, phenolic glycosides, and androquinone glycosides, saponin glycosides. So, there are 4 types of glycosides. So, from cardiac glycoside, we are getting digoxin. From phenolic glycosides, salicylene. From androquinone glycosides, senosides A and B. From saponin glycoside, diosine. So, these are all sources of this glycosides. And in this glycoside, there are 2 parts. Sugar, aglycone part is a sugar, glycone part is a sugar part, and aglycone is a non-sugar part. So, in this digoxin, it's containing, so you know the glycosides means it's containing sugar part and non-sugar part. Sugar is the glycone part, non-sugar is the aglycone part. So, digitoxose is a glycone part, digoxin is a aglycone part, and glucose, and in salicylene, glucose is a glycone part, salicyligenin is a aglycone part, and in senoside A and B, glucose part is glucose, that means sugar part is glucose, and non-sugar part is androquinone derivatives, and diazosin is a glucose plus ramenose and diazogenin. So, it is uses of this digoxin, the glycoside, is increases the force of the heart contractions, and salicyligenin is a uses is analgesic and antipyretic. Androquinone is a laxative and diazogenin is used in the synthesis of steroidal drugs. Next, biosynthesis of glycosides is a key reactions we'll discuss. So, for first step, formation of sugar moiety, that means glycone path. So, in this glucose, first is uridine diphosphate to glucose. It's activated sugar donor used for glycosylation reactions. And in this reaction, you look over this reaction, glucose in the presence of ATP and hexokinase, it converted into glucose 6-phosphate. Then glucose 6-phosphate in the presence of phosphoglutamate enzymes, it converted into glucose 1-phosphate. Then this glucose 1-phosphate, it combined with the uridine triphosphate in the presence of uridine diphosphate and glucose pyrophosphorylase to convert into uridine diphosphate glucose and PPI. So, in this is step 1. So, formation of sugar moiety, that means glycone path. So, uridine diphosphate glucose is formed. The step 2, the interconversion of activated sugar. So, here to provide structural variety of sugars like glucose, galactose, rhamnose, etc. in the plant glycosides. So, first step, uridine diphosphate glucose is formed. So, that uridine diphosphate glucose in the presence of epimerase enzymes, it's converted into uridine diphosphate galactose. So, then this uridine diphosphate galactose glucose in the presence of dehydratase and reductase enzymes, it converted into uridine diphosphate rhamnose. So, first is converted into galactose in presence of epimerase, and second is converted into uridine diphosphate rhamnose in presence of dehydratase and reductase enzymes. Then this GDP mannose is converted into, in the presence of epimerase enzyme, is converted into guanine diphosphate fucose. That means fructose. So, guanine diphosphate. Next, step 3, biosynthesis of glycosides and key reactions. So, there is a third step, formation of A-glycone, that means non-sugar part. So, the first previous slide we have discussed about the formation of glycone part, it's a sugar part. So, now from the step 3 onwards, we are going to discuss about A-glycone part. So, formation of A-glycone, that means non-sugar part. A-glycones are derived from secondary metabolic pathway such as cyclic acid pathway, acetate malonate pathway, mevalonate pathway and phenyl propanoid pathway. So, from this step, going to form the A-glycone part. So, in cyclic acid pathway, the condensation of pyroenol pyruvate and erythrose 4-phosphate, it form the cyclic acid. Then this cyclic acid is converting into the aromatic rings, and it leads. So, examples of this, phenolic glycoside is formed. So, you read this slide carefully. So, there are many MCQs they may ask from this slide. You take a snapshot and read it. Next, biosynthesis of glycosides. The step 4, step 3 is the formation of A-glycone part. That means non-sugar part is formed from the cyclic acid pathway. So, now, and fourth step, glycosylation reaction is a key step for the formation of glycosides. So, this is the final conjugation step, joining sugar and A-glycone, non-sugar part. So, A-glycone hydroxides containing OH group and uridine diphosphate glucose. So, it is in the presence of glycoside transferase enzymes. It's converted into A-glycone linked with oxygen and glycosides. So it's a O-glycosidic linkage is formed. And uridine diphosphate. So it's the formation of glycosides. And fifth step, types of glycosidic linkage formed. O-glycoside, it means sugar O connected with O A-glycone part. That means oxygen A-glycone part. C-glycosides means directly carbon-carbon double bond is formed. So example, arbutin rutin is a O-glycoside part of O-glycosides. And C-glycoside means directly carbon-carbon bond is formed. Aloin. And N-glycosides means sugar moiety connected with N of A-glycone. And its example is adenosine purine nucleosides. S-glycosides, sugar S connected cyanide group is connected with A-glycone. So cinegrine and mustard oil glycosides. So already we have discussed this. Step 1, step 2, step 3, step 4, step 5. Step 1 is the formation of sugar moiety. Step 2 is the interconversion of activated sugar. And step 3 is the formation of A-glycone, that means non-sugar part. And step 4 is the glycosylation reaction to the formation of final glycosides. And final step, types of glycosidic linkage formed. So OCN, there are 4 glycosidic linkages are there. So you take snapshot of this slides and you read it. So regulations and compartmentalization in biosynthesis of glycosides. So first, locations, the glycosides where it is takes place means glycoside biosynthesis occurs in the cytosol or endoplasmic reticulum of the plant. And enzyme specificity, the plant tissues express unique glycosyl transferase leading to structural diversity. And where it is stored, storage, glycosides accumulate in vacuole and separated from hydrolytic enzymes, that means glycosidase enzymes. So next we will move into the questions. Important questions. It's very important for the GPAT and MRB and drug inspector. The molecules found in all plants and necessary for the life of the plants are called as. So the answer is option B, secondary metabolites. So the molecules found in all plants and necessary for the life of the plants are called as, it is called secondary metabolites. Next, the sugar part of the glycosides is activated for glycosylation in which form? So sugar part of the glycosides or glycone is activated for glycosylation in which form? So answer is option C, uridine diphosphate glucose. So uridine diphosphate glucose act as the activated sugar donor during glycoside formation and catalyzed by glucosyl transferase enzyme. During glycosides biosynthesis, the sugar donor uridine diphosphate glucose is formed from, so the answer is option B, glucose 1-phosphate and uridine triphosphate. So glucose 1-phosphate and uridine triphosphate is synthesized by the reactions of glucose 1-phosphate with uridine triphosphate and catalyzed by uridine diphosphate glucose pyrophosphopyrolase enzyme. The universal sugar donor for plant secondary metabolites glycosylation is, so the answer is option B, uridine diphosphate glucose. So uridine diphosphate glucose is most common activated sugar donor used by glycosyl transferase enzymes during glycoside synthesis. The first step in glycoside biosynthesis involves, so option A, activation of sugar. So the first step means activation of sugar, that means formation of sugar part, glycan part. So sugar moiety is the first activated by uridine diphosphate glucose before it participate in glycosidic formation. So the first is the formation of activation of sugar, formation of sugar part. The enzyme responsible for the transfer of sugar moiety to the aglycone is option B, answer, glycosyl transferase. So glycosyl transferase enzymes the transfer of sugar from uridine diphosphate sugar to the aglycone part and forming the glycosidic bond. Like which cofactor is commonly required for glycosyl transferase enzyme activity? So the answer is option D, none of the above. So these are all not involving in the glycosyl transferase enzyme activity. So glycosyl transferase enzyme activity uses activated nucleotide sugars like uridine diphosphate glucose. So not in the UDP and ATP. It is UDP, this glucose part is required as a substrate and do not require cofactor like ATP and adenine diphosphate for catalase. NADPH. The biological activity of glycoside mainly depends on option B is the answer, the aglycone part. So the aglycone is a genin determines the pharmacological activity while the sugar affects solubility and transportation. So sugar parts mainly is used for the solubility and transportation of the active metabolites. The sugar moiety in most plant glycosidic is attracted to the aglycone through which type of bond? So the answer is option B, glycosidic bond. So in glycosides, the sugar, that means aglycone part and non-sugar aglycone parts are linked via glucosidic bond and this bond is formed from enzymatically by glucose transferase enzyme. The purpose of glycosylation in glycoside biosynthesis is to, so option B is the answer, so increase solubility and stability. So glycosylation enhances the solubility, stability and transport of secondary metabolites within plants. Which of the following statement about glycoside biosynthesis is true? So you read this sentence. Option A, the aglycone is always synthesized after sugar activation. So which of the following statement about glycoside biosynthesis is true? So option B, both the sugar and aglycone are independently formed and later conjugated. Option B, C, glycosidic bond form spontaneously without enzymes. Option D, sugar formation occurs only in root. So among this, so option B statement is correct. So the answer is option B. So both sugar and aglycones are independently formed. So the first step sugar form, the second step aglycone, so non-sugar will form, then both are conjugated. The aglycone part and glycone part is glycone part is from carbohydrate metabolisms formed and aglycone part is formed from secondary metabolic pathway, and after formations both are formed separately and joined by enzymatic glycosylation reaction. The aglycone part of the cardiac glycoside is biosynthesized through which pathway? So the answer is option B, mevalonate pathway. The steroidal nucleus of cardiac glycosides is derived from the mevalonate, so isoprenoid pathway. Which of the following is a a phenolic glycoside formed via the cyclic acid pathway? So option A, arbutin. So arbutin is a hydroquinone combined with glucose. Is a phenolic glycoside synthesized via cyclic pathway. Which of the following is derived from the acetate pathway? Polypeptide pathway. So the answer is option B, aloin. So aloin is type of anthraquinone glycosides which are synthesized through the acetate malonate pathway. So very important. So it is synthesized from acetate malonate pathway. In plant, the rhamnose used in glycosides formation is obtained from option A, glucose via uridine diphosphate rhamnose. So rhamnose is biosynthesized from glucose through uridine diphosphate glucose converted into uridine diphosphate rhamnose. So by the reduction and epimerization reactions. Anthraquinone glycosides like imodine and aloin are formed through which biosynthetic route? So option B, acetate malonate pathway. So anthraquinones are polypeptide and synthesized through acetate malonate pathway using acetyl-CoA and malonyl-CoA as a precursors. Which of the following correctly pairs of glucosides with its source and biosynthetic class? So the answer is option A, salicylene. So willow bark phenolic glycosides. So salicylenes from Salicylic alba is a willow bark. Is a phenolic glycosides derived from cyclic pathway and has analgesic and antipyretic properties. The biosynthesis of saponin glycosides involves which precursors pathway for aglycone part? So the answer is option B, mevalonate pathway. So the saponin possesses a triterpenoid or steroidal aglycone which originate from the mevalonate isoprenoid pathway. Which of the following glycosides is flavonoids in nature and derived through the phenylpropanoid pathway? So option A, rutin. So rutin is a flavonoid glycosides synthesized via phenylpropanoid pathway from phenylalanine through cinnamic acid intermediates. The primary role of the sugar moiety in a glycoside molecule is to, so answer in the comment box. You join in our WhatsApp community, you will get number of test series and hands out materials. Thank you. Like, subscribe and share. Thank you.